Welcome back to the deep dive. Today, we're really getting into the nitty-gritty of microbial ecology. We're trying to tackle this idea that pure culture is, well, basically a lab invention.
That's absolutely the core idea. Because in reality, any organism you look at, a plant, an animal, you yourself, you're not just an individual. You're this complex, co-evolved ecosystem.
Right, a kind of biological collective.
Exactly. We use terms like metaorganism or holobiont. Fancy words, maybe, but they capture that essential idea. Life is often a partnership.
So our mission today is to kind of map out this world, give everyone the tools to understand it.
Precisely. We'll explore the 7 main ways microbes interact, from helping each other out, mutualism, all the way to outright warfare like predation.
Okay, let's dive right in. And we're starting small, right? With the freshwater polyp, Hydra.
Yeah, Hydra is a fantastic model. It's relatively simple anatomically, but getting its microbial community established, that's surprisingly complex, almost like its own development, its own embryogenesis.
And it's not just passively accepting microbes, is it? It's controlling the situation.
Oh, absolutely. The host, the Hydra, is actively managing things. We see it producing these antimicrobial peptides, AMPs, which are like little chemical gatekeepers deciding who gets to settle down.
And what I found really neat was that it doesn't always need specialized tools. It uses its own stuff, right? Like that neuropeptide, NDA1.
That's the fascinating part. NDA1 helps build the Hydra's nervous system, but it also acts as an AMP.
So it's doing double duty.
Exactly. It actually helps structure where the main bacterium, Curvybacter, lives on the Hydra. The host is literally using its own developmental signals to, landscape its microbiome.
Wow, and the control doesn't stop there. It even gets into how bacteria talk to each other, this quorum sensing thing.
Right. Bacteria use these chemical signals, N-acyl homoserine lactones, or EHLs, to coordinate what the whole population does. Think of it like taking a vote chemically.
But the Hydra intercepts the message.
It does more than intercept. It produces an enzyme, an oxidoreductase, that chemically changes the AHL molecule.
Whoa, okay. So it's not just listening, it's rewriting the conversation.
You got it. It allows the host to influence what genes the bacteria turn on, what they do, their phenotype, without just killing them off. It picks its residents, then manages their behavior.
That really sets the scene. Control is key. So to understand why this control evolved, we need the full picture, these 7 interaction types. Maybe we can think of it like a scorecard. Partner A, partner B, win, lose, or draw.
That's a great way to visualize it, like figure 27.1 in the text. Let's start with the positive stuff. Mutualism, that's the top tier. Obligatory, both partners win, plus plus. They literally can't live without each other.
Okay, and slightly less intense.
That's cooperation. Still win-win, plus plus, but it's not obligatory. They do better together, sure, but they can survive apart if they have to.
Got it. Then there's the neutral one, commensalism. One benefits, the other is just there, plus zero.
Basically. One partner gets something out of it, the other, completely unaffected. We used to think our own gut microbes were mostly like this.
Yeah, but we know better now, right? They're definitely not just passive writers.
Definitely not. Okay, moving to the negative side. We have predation, straightforward killing and eating, plus slaying. One benefits the other. Well, doesn't.
And similar but different is parasitism. Also plus flank, but it's more of a long game.
Right. The parasite lives on or in the host, often keeping it alive at least for a while, so the parasite can reproduce and spread. It's exploitation, but sustained.
Okay, 2 more. Amensalism. Sounds a bit weird. 0. One organism hurts another, often with chemicals, but the producer doesn't really get anything out of it.
Think of it like collateral damage almost. Or maybe just making your environment hostile. One releases something harmful, the other suffers. The first one is unaffected by that specific interaction.
Like microbial chemical warfare without a clear strategic goal for the attacker.
Sort of, yeah. And finally, competition. The classic. Everyone loses, or at least does worse, because they're fighting over the same limited resources, like food or space. This leads to that idea of competitive exclusion.
Okay, so that's the spectrum. But regardless of the type of relationship, these microbes, these symbionts, they all have to do certain things, right? Get in, stay in, reproduce.
Absolutely. They need to colonize the host, reproduce successfully, persist within that environment, and then, crucially, transmit to a new host.
And that transmission can be sideways, like catching something from the environment horizontal.
Or passed down directly from parent to child, vertical transmission.
And the more interdependent they become, the more we need these other terms, like Metaorganism, holobiont.
Exactly. Because it stops being just a host and a symbiont and starts becoming this single integrated life unit. They're fundamentally intertwined.
Speaking of intertwine, let's look at that ultimate commitment, obligate mutualism. The classic example is the aphid and Buchnera.
Ah, yes, Buchnera aphidicola, a partnership going back maybe 150 million years. Aphids drink plant sap, which is like sugary water, basically. Very poor in essential amino acids.
Stuff the aphid can't make itself.
Right, it's missing 10 essential ones. So Buchnera, living safe inside special aphid cells, synthesizes exactly those 10 amino acids.
Yeah.
The aphid provides the home and basic nutrients. Buchnera provides the missing essentials.
And without Buchnera.
The aphid dies. It's completely dependent.
But this dependence comes at a cost for the bacterium, doesn't it? This genomic reduction thing.
A huge cost. Because the aphid handles so much finding food, protection Buchnera just doesn't need all the genes its ancestors had. So over millions of years, its genome is shrunk dramatically, down to about 0.65 megabases. It's lost the ability to do many things for itself.
It's locked in. It can't survive outside the aphid anymore.
Totally locked in. An obligate mutualist. This kind of extreme specialization happens elsewhere, too. Think about the termite gut.
Ah, the wood eaters. That's going to be a complex system inside.
Well, it's incredible. A multi-partner mutualism. The termite needs to break down tough wood, lignocellulose, and it also needs nitrogen, which is scarce in wood.
So multiple problems, multiple microbial solutions.
Exactly. Nitrogen-fixing bacteria provide the organic nitrogen, but breaking down the wood, that takes a team. Large protists, single-celled eukaryotes, do the heavy lifting. They have these special organelles called hydrogenosomes and ferment the cellulose.
Into what? What does the termite actually use?
They turn it into acetate, CO2, and hydrogen gas, H2. And that acetate, that's the termite's main source of carbon and energy.
But you said multi-partner. There's more.
There is. It gets even more nested. Some of those protists, like Trichonympha, actually have their own bacterial endosymbionts living inside them.
Wait, bacteria living inside protists living inside a termite.
Yep, an endosymbiont of an endosymbiont. These internal bacteria help the protist with things like amino acid conversions. It's like microscopic Russian dolls all working together.
That's wild. Okay, for extreme mutualism, what about those deep-sea vents, the Riftia tube worms? No mouth, no gut, living in toxic soup.
Right, thousands of meters down. Pitch black, crushing pressure, water blasting out at hundreds of degrees Celsius, loaded with hydrogen sulfide. It's about as extreme as it gets.
So how do they eat?
They don't. Not in the way we think. They rely completely on chemosynthetic bacteria. Candidatus and Riftia percephin packed into a special organ called the trophosome.
Chemosynthetic, meaning they make food from chemicals, not light.
Exactly. They use the chemical energy from hydrogen sulfide, H2S. The big challenge is getting the fuel, H2S, and the oxidant, oxygen, O2, to the bacteria safely. These two chemicals react readily.
So how does the worm manage that?
It has this amazing bright red hemoglobin. Unlike ours, which just carries oxygen, the Riftia hemoglobin is specially designed to bind and transport both H2S and O2 deep into the trophosome without them reacting prematurely.
Wow, like a specialized delivery truck for dangerous goods.
Kind of, yeah. The bacteria then take the H2S and O2, use the energy released from reacting them, and use that energy to fix carbon dioxide into organic matter using the Calvin-Benson cycle. They make sugars in total darkness.
And that's what feeds the worm. Incredible.
It really is, and it highlights how life finds a way, often through partnerships. Another metabolic powerhouse driven by microbes is the rumen.
Ah, the cow's stomach. Or stomachs, really.
Right. Ruminants like cows, sheep, goats, they have this huge anaerobic fermentation chamber, the rumen. It's packed with microbes, maybe 10 to the 12th organisms per milliliter. It's a world unto itself.
All breaking down tough plant material, grass and hay.
Exactly. Stuff the animal can't digest directly. The microbes break it down into short-chain fatty acids, acetate, butyrate, propionate. Those are the actual energy source for the cow.
But you mentioned thermodynamics earlier. Sometimes these breakdown reactions are tricky.
Very tricky, especially breaking down some fatty acids further. The reactions often produce hydrogen gas, H2. If that H2 builds up, the reaction becomes energetically unfavorable. It requires energy input, a positive delta G, so it stops.
So the whole system would grind to a halt.
It could. This is where syntrophy comes in, literally eating together. And it often involves interspecies hydrogen transfer, or IHT.
Meaning someone else uses the hydrogen.
Precisely. You have methanogenic archaea methane producers. They gobble up the H2, combine it with CO2, and make methane, CH4. By keeping the H2 levels super low, they make the original fatty acid breakdown reaction thermodynamically favorable. They change the delta G from positive to negative.
So the methanogens act like garbage disposal for hydrogen, allowing the fermenters to keep working.
That's a perfect analogy.
Yeah.
It pulls the reaction forward. And sometimes they get really close to ensure this works well.
Suppose how?
We've seen bacteria like Peletomaculum literally tethering themselves to methanogens like Methanothermobacter. They use a modified flagellum, like a little grappling hook, to stay physically connected.
Wow. A physical link for efficient chemical exchange. That's amazing cooperation.
It is. And that idea of cooperation, which isn't strictly necessary for survival, bridges us nicely into other types of interactions.
Right, like the difference between that obligate mutualism in aphids and something more flexible. You mentioned cooperation earlier. Plus but non-obligatory.
Yeah, a good example is the bacterium Xenorhabdus and its nematode partner, Steinernema. They can technically survive apart, but together they're a deadly team for insects.
How does that work?
The nematode carries the bacteria in its gut. It infects an insect larva, then releases the Xenorhabdus. The bacteria multiply rapidly and produce toxins that kill the insect fast.
Okay, so the nematode provides transport, the bacteria do the killing. What else?
The bacteria also pump out antimicrobials to keep other microbes away, preserving the insect carcass as food for the nematode. And then bacterial signals trigger the nematode to develop and reproduce inside the dead insect. It's a coordinated attack and exploitation.
So highly beneficial, but they could technically go it alone, just less effectively.
Exactly. Now, commensalism plus zero is often about environment modification. Think about biofilms.
The slimy layers microbes form.
Right. The first microbes to land on a surface might change it. Maybe they produce some sticky polymers. That makes it easier for other microbes to attach later. The first ones might not benefit from the latecomers, but the latecomers definitely benefit from the pioneers.
Or like a fermentation. One microbe makes acid waste.
And that acid creates a perfect niche for an acid-loving microbe to thrive. The first one benefits, gets rid of waste. The second benefits, gets a good environment. But maybe the first one doesn't directly gain from the second's presence, or is unharmed, hence the zero.
Which, as you said, is why we've reevaluated our own gut microbes. They do way too much for us to be just commensals.
Absolutely. They're training our immune system, providing vitamins, keeping pathogens out. That sounds much more like mutualism.
Okay, let's shift gears to the more antagonistic side. Wolbachia. You called it a master manipulator.
Oh, Wolbachia is incredible. It's a type of rickettsial bacterium that infects a huge number of insects and other arthropods. And its success is all about manipulating host reproduction.
How does it do that?
It uses several tricks to ensure it gets passed down through the female egg cells, that vertical transmission. It can cause cytoplasmic incompatibility, where infected males can only successfully reproduce with infected females.
So uninfected females are at a disadvantage.
Right. Or it can kill off male embryos entirely, or even turn genetic males into functional females. All strategies maximize the number of infected females passing Wolbachia to the next generation.
That's some serious evolutionary warfare.
It is. But what's really interesting now is we're using it for our benefit.
Oh, how?
It turns out Wolbachia infection makes it harder for certain viruses, like dengue and Zika, to replicate inside mosquitoes. So scientists are releasing Wolbachia-infected Aedes mosquitoes.
And they spread the Wolbachia, and this blocks disease transmission to humans.
That's the goal, yeah. Biological control, using a parasite's own manipulative tricks against disease vectors. Pretty clever.
Very clever. Okay, let's talk amensalism, the chemical warfare one, the teen ants and their fungi.
Ah, the leafcutter ants and their relatives. This is a classic multi-layered symbiosis. The ants cultivate a specific fungus, Leucocaprinus, in underground gardens. That's their food.
Like little farmers.
Exactly. But their fungal garden is constantly under attack by another fungus, a parasite called Xenorhabdus.
So the ants need garden security.
They do, and they get it from bacteria. They carry bacteria, usually pseudonocardia, in special structures, little crypts on their bodies. These bacteria produce potent antifungal compounds that kill the Xenorhabdus parasite.
Okay, so ants farm fungus, bacteria protect fungus. That's 3 partners. You mentioned 4 or 5.
Right. This is where it gets even cooler. Making those antifungals is costly for the pseudonocardia.
Yeah.
So there's evolutionary pressure for cheater bacteria to arise, ones that live on the ant but don't make the antifungal, saving energy.
How does the system deal with cheaters?
Enter potentially a 5th player, a black yeast, Phaeolophora. This yeast seems to prey specifically on the pseudonocardia, but evidence suggests it targets the cheaters, the bacteria not producing the helpful antifungal.
So the yeast acts like quality control, policing the bacteria to make sure they pull their weight.
That's the hypothesis. It maintains the integrity of the protective bacterial symbiosis by eliminating the non-producers. It's an incredibly complex web of interactions.
Mind-boggling. Okay, quick distinction. Predation versus parasitism. Both are plus D.
The key difference is outcome for the prey host.
Yeah.
Predation, death. The predator kills the prey for nutrients. Think Vampyrococcus latching onto the outside of its prey, or Daptobacter invading, or Mysococcus swarming and digesting other bacteria.
Whereas parasitism excludes coexistence, at least for a while, like Mycobacterium leprae, causing leprosy. It lives within the host long term.
Exactly, though sometimes the lines blur. Look at lichens.
The crusty stuff on rocks and trees, fungus plus algae.
Or fungus plus cyanobacteria. It looks like a perfect mutualism. The fungus provides structure, water retention, minerals. The photosynthetic partner, phycobiont, provides sugars from photosynthesis.
Seems win-win.
It does. But often, if you culture the algal or cyanobacterial partners separately in the lab, it grows much faster and healthier on its own than it does inside the lichen.
Oh, so the fungus might actually be holding it back.
It suggests the fungus is controlling the phycobiont's growth and essentially farming it, taking most of the photosynthetic products. So many biologists classify it as a very controlled, stable form of parasitism plus make, rather than true mutualism.
Huh. Interesting perspective shift. Okay, last one. Competition. Fighting for resources.
Right. And this is fundamentally important in places like our gut. The competitive exclusion principle basically says if two species need the exact same limited resource, one will eventually outcompete the other.
And this helps protect us.
It's a key part of colonization resistance. Your established gut microbes are already using up the readily available nutrients and occupying the space. This makes it really hard for an incoming pathogen to find a foothold and establish itself. They get outcompeted.
So wrapping this all up, we started saying pure culture is a myth. Everything is connected.
Pretty much. The world, biologically speaking, operates as a holobiont. These intricate relationships, especially the metabolic handoffs, like the hydrogen transfer in the rumen making reactions possible, or the Riftia worm delivering sulfide and oxygen, they drive entire ecosystems.
And the complexity is just stunning, from bacteria physically linking up to fungi policing bacteria to hosts rewriting bacterial communication.
It's an incredibly dynamic and complex world down there.
So we talked about that extreme dependence, like Buchnera losing its genes and being totally reliant on the aphid. It makes you think about our own microbiome, which we now see as mutualistic.
Exactly. We depend on them for functions we may not even fully understand yet.
So here's the final thought for you, the listener. Given this deep, co-evolved dependence, what happens when we, the host, do something drastic, like taking a broad-spectrum antibiotic that wipes out not just pathogens, but also potentially crucial microbial partners?
Partners that might have lost some of their own functions over evolutionary time, relying on us, just as we rely on them. What non-obvious essential functions might we be losing, potentially permanently altering our own biology when we disrupt that ancient partnership? Think about things like recurrent yeast infections after antibiotics, a visible disruption. What else might be happening unseen?
A sobering reminder that we're not just individuals, but ecosystems. Definitely something to think about. Thanks for joining us on The Deep Dive.